The aim of this procedure is to prepare antigen-protected functional red blood cells. This is accomplished by first preparing the HPG polymer. In the second step, the HPG is functionalized through the addition of succinimidyl succinate groups. Next, whole blood from healthy donors is washed with saline to separate out the red blood cells. In the final step, the isolated red blood cells are treated with the functionalized HPG. Ultimately, the protection of the surface antigens on the modified cells by the functionalized HPG is assayed by flow cytometry.
The main advantage of our technique compared to existing methods is that HPG conjugations yield functional red blood cells that circulate normally.
The implication of this technique extends toward chronic blood transfusion, as HPG-modified red blood cells have reduced antigenicity and normal circulation in vivo.
Demonstrating the procedure will be Dr. Rafi Chapanian, a postdoctoral fellow, and Iren Constantinescu, a technician in my laboratory.
Three days before collecting whole blood from consented healthy donors, dry lyophilized HPG in a round bottom flask overnight under vacuum at 90 degrees Celsius. The next morning, remove the flask from the oil bath and allow it to reach room temperature, and then dissolve the dried HPG in 2 ml of anhydrous pyridine. Next, add a catalytic amount of dimethylamino pyridine